Hi, I am Dr. P.R. Padma, Professor in the Department of Biochemistry, Biotechnology and Bioinformatics of Avinashilingam University for Women, Coimbatore. You have now chosen to study the module on degradation of nucleic acids, which is a part of Nutritional Biochemistry paper in the subject Food Science Nutrition. Nucleic acid metabolism is the process by which nucleic acids, namely DNA and RNA, are synthesized and degraded. Nucleic acids are polymers of nucleotides. Nucleotide synthesis is an anabolic mechanism generally involving the chemical reaction of phosphate, pentose sugar and nitrogenous base. The destruction of nucleic acid is a catabolic reaction. Both the synthesis and degradation reactions require enzymes to facilitate the events. Any defects or deficiencies in these enzymes can lead to a variety of diseases. To learn the details of nucleic acid degradation with special reference to the purine catabolic pathway and pyrimidine catabolic pathway, and also get an introduction about the diseases and disorders that result from disturbances in these pathways. Let us now do a quick revision of the concepts of the structure of nucleic acids. Nucleic acid is a complex organic substance present in living cells, especially DNA, that is deoxyribonucleic acid, or RNA, that is ribonucleic acid. These molecules consist of many nucleotides which form the monomers of these and linked in a long chain. These monomers are called nucleotides. Each nucleotide has three components: a 5-carbon sugar, a pentose sugar as we call it, a phosphate group and a nitrogenous base. If the sugar is deoxyribose, the polymer is DNA or deoxyribonucleic acid, and if the sugar is ribose, the polymer is RNA or ribonucleic acid. The nitrogen containing bases belong to two families of compounds, namely purines and pyrimidines. Both DNA and RNA contain the same purine bases, namely adenine and guanine, denoted by capital A and capital G. Both DNA and RNA also contain the pyrimidine cytosine, denoted as capital C, but they differ in their second pyrimidine base, that is DNA contains thymine, denoted by capital T, and RNA contains uracil, denoted by capital U. A double-ringed crystalline organic base, which is not known to occur naturally, from which is derived the nitrogen bases adenine and guanine, which constitute the purines, as well as uric acid, which is a metabolic end product of purine degradation, are very important as far as the metabolism of purines are concerned. Purines consist of a 6-membered and a 5-membered nitrogen containing rings fused together. Pyrimidines are single ringed crystalline organic bases that form uracil, cytosine or thymine, and the parent compound of many drugs including barbiturates and other such painkillers. Pyrimidines have a single 6-membered nitrogen containing ring. Having refreshed the concepts that we learned in the earlier module, let us now consider the catabolism of nucleic acids. Nucleases and nucleotidases degrade nucleic acid chains down to free nucleotides or the monomers, and then to nucleosides by the removal of phosphate groups. The purine nucleosides are then degraded further into uric acid, which is excreted in the urine. The purine nucleosides and free bases are then deaminated, that is the removal of amino group, to form hypoxanthine and xanthine by various specific deaminases. These enzymes include AMP aminohydrolase, otherwise called adenylic deaminase, adenosine deaminase, adenine deaminase and guanine deaminase, among which the adenine deaminase is present only in trace amounts in animal tissues, while all the others are widely distributed in all animal tissues. In the presence of xanthine oxidase, hypoxanthine and xanthine are converted to uric acid, which is subsequently converted to allantoin by uricase. Xanthin oxidase is present in most animal tissues, but its distribution varies between tissue to tissue and between the animal species. However, it appears to be present at high levels in the liver of all animals. Thereby, all animals are very capable of carrying out purine degradation. In most mammals, allantoin is the final product of purine degradation chain and is excreted in the urine as the major component of the purine end products. Hypoxanthine, xanthin and uric acid are also excreted, probably due to their high clearance rate from the blood. Man and other primates excrete uric acid as the final product of purine catabolism as the result of lacking uricase. Birds also lack uricase and therefore excrete uric acid. The major advantage of doing so in birds is saving water. However, uric acid can also be produced from urea in birds. Other species of less developed animals like fish have enzymes to further degrade allantoin to allantoic acid and glyoxylic acid and finally to urea, which is meant for excretion. The major steps involved in this degradation pathway are depicted in the scheme that is now displayed on your screen. I urge you to take a minute to go through the reactions so that your understanding of the process becomes clearer. As you can see in the scheme of reactions, adenosine first deaminates to inosine, then cleaves to form hypoxanthine, which in turn oxidizes to xanthin and then uric acid. Guanosine first cleaves to release guanine, then deaminates to become xanthin and then oxidizes to form uric acid as before. In general, the nucleotide monophosphates, namely AMP, IMP and CMP, are converted to their respective nucleoside forms, that is adenosine, inosine and guanosine, by the action of nucleotidase. The amino group either from AMP or adenosine can be removed to produce IMP or inosine respectively. Inosine and guanosine are respectively converted to hypoxanthine and guanine, both of which are purine bases. This is done by purine nucleoside phosphorylase. Adenosine is not degraded by this enzyme and therefore it has to be converted to inosine. Guanine undergoes deamination by guanase to form xanthin. Xanthin oxidase is an important enzyme that converts hypoxanthine to xanthin first and then converts xanthin to uric acid. This enzyme contains FAD, molybdenum and iron and is exclusively found in the liver and small intestine of animals. Xanthin oxidase liberates hydrogen peroxide, which is harmful to the tissues when not scavenged. Catalase is an enzyme that can effectively cleave hydrogen peroxide to water and oxygen. When reactions of purine degradation do not occur sequentially, several disorders can ensue. We shall now consider the major disorders of purine degradation pathway. Let's start with gout. Gout is a metabolic disease associated with the overproduction of uric acid. At the physiological pH, uric acid is found in a more soluble form as sodium urate. In severe hyperuricemia, crystals of sodium urate get deposited in the soft tissues, particularly in the joints. Such deposits are commonly called as tophi. This causes inflammation in the joints, resulting in a painful gouty arthritis. Sodium urate and uric acid may also precipitate in the kidneys and ureters, resulting in renal damage and kidney stone formation. Historically, gout was found to be often associated with high living, overeating and alcohol consumption. In the earlier days, alcohol was contaminated with lead during its manufacture and storage. Lead poisoning leads to kidney damage and decreased uric acid excretion, causing gout. Thus, Gout is caused by an elevated uric acid level in the bloodstream and the accumulation of uric acid crystals. Uric acid crystals deposit in the joints, causing inflammation of the joint, leading to pain, redness, heat, and swelling. Causes of an elevated blood uric acid level, which is referred to as hyperuricemia, include genetics, obesity, certain medications such as diuretics or water pills as they call it, and chronic decreased kidney function. When gout is mild, infrequent and uncomplicated, it can be treated with diet and lifestyle changes alone. But when attacks are more frequent, or when uric acid kidney stones have occurred, or tophi are present, or there is evidence of joint damage from gout attacks, medications are typically used to lower the blood level of uric acid. The drug of choice for the treatment of primary gout is allopurinol, which is a structural analog of hypoxanthine and therefore can competitively inhibit the enzyme xanthine oxidase. Inhibition of xanthine oxidase by allopurinol leads to the accumulation of hypoxanthine and xanthine. These two compounds are more soluble than uric acid and therefore can be easily excreted, thus reducing gout attacks. Besides the drug therapy, restriction in dietary intake of purines and alcohol is also advised. Consumption of plenty of water will also be useful because it washes out the uric acid and does not allow it to deposit. The anti-inflammatory drug colchicine is also used for treatment of gouty arthritis. Other anti-inflammatory drugs such as phenylbutazone, indomethacin, oxyphenybutazone, corticosteroids are also useful. Let's move on to the next condition, SCID or severe combined immunodeficiency as we call it. This condition, SCID, represents a group of rare, sometimes fatal congenital disorders characterized by little or no immune response. The deficiency of an enzyme called adenosine deaminase, which is abbreviated as ADA, causes severe combined immunodeficiency or SCID. This involves T cell and usually the B cell dysfunction. T cells and B cells are important components of the immune system. It is explained that ADA deficiency results in the accumulation of DATP or deoxy-ATP, which is an inhibitor of ribonucleotide reductase, and therefore DNA synthesis and cell replication are affected. The deficiency of purine nucleotide phosphorylase is associated with impairment of T cell function but has no effect on B cell function. Uric acid synthesis is decreased and the tissue levels of purine nucleotides and nucleosides are higher. It is believed that deoxy-GTP inhibits the development of normal T cells. The most common treatment for SCID is bone marrow transplantation, which has been successful using either a matched, related or unrelated donor or even a half-matched donor, which could be either of the parents. The half-matched type of transplant is called haploidentical. Haploidentical bone marrow transplants require the donor marrow to be depleted of all mature T cells to avoid the occurrence of graft versus host disease or DVHD. Consequently, a functional immune system takes longer to develop in the patient who receives a haploidentical bone marrow transplant compared to a patient receiving a matched transplant. Nowadays, transplants done in the first 3 months of life have a high success rate. Physicians have also done before the child is born and also by using cord blood, which is rich in stem cells. In utero transplants allow the fetus to develop a functional immune system in the sterile environment of the uterus. However, complications like GVHD would be difficult to detect or treat if they were to occur in utero. More recently, gene therapy has been attempted as an alternative to the bone marrow transplant. transduction of the missing gene to hematopoietic stem cells using viral vectors is also being tested in ADA skid and X-linked skid. While this has been very successful in some patients, others have developed serious complications from this treatment. More research is needed to understand these reactions involved and arrive at a good treatment strategy. There are also some non-curative methods for treating skid. This means the patient is not treated to cure the disease, but to keep the patient comfortable and free of the complications of the disease as much as possible. Among such methods, reverse isolation involves the use of laminar airflow and mechanical barriers to isolate the patient from any harmful pathogens present in the external environment. With ADA skid and enzyme replacement therapy, this is needed. Enzyme replacement therapy is done wherein the patient is injected with polyethylene glycol coupled adenosine deaminase, which is abbreviated as PEG-ADA. This metabolizes the toxic substrates of ADA enzyme and prevents their accumulation. Treatment with PEG-ADA may be used to restore T cell function for a short term, enough to clear up any existing infections before proceeding with a curative treatment like bone marrow transplant. The next condition that is associated with purine degradation is hypouricemia. Hypouricemia is a condition that results from below normal levels of uric acid in the blood. That is, a uric acid level of less than 2 mg per 100 ml in the serum represents hypouricemia. This condition is mostly associated with a rare genetic defect in the enzyme xanthine oxidase. It leads to increased excretion of xanthine and hypoxanthine. Xanthinuria frequently causes the formation of xanthine stones in the urinary tract. Decreased uric acid production can be caused by several rare inherited disorders of purine synthesis and catabolism, and more commonly by acquired deficiency of xanthine oxidase due to allopurinol therapy or liver disease. Other than these disorders and diseases, several inherited disorders associated with purine metabolism are also known to occur. Inherited disorders resulting in decreased uric acid production include hereditary xanthinuria and purine nucleosidase phosphorylase deficiency. Now, let us move on to the catabolism of pyrimidine nucleotides. In contrast to the purines, pyrimidines undergo ring cleavage, and the usual end products of catabolism are beta amino acids. Ammonia and carbon dioxide are also produced. Pyrimidines from nucleic acids or the energy pool are acted upon by nucleotidases and pyrimidine nucleoside phosphorylase to yield the free bases. The 4-amino group of both cytosine and 5-methyl cytosine, that is thymine, is released as ammonia. Beta alanine can be recycled into the synthesis of coenzyme A. Catabolism of the pyrimidine base thymine or 5-methyl uracil yields beta amino isobutyric acid instead of beta alanine. Pathways presented thus far in this chapter account for the synthesis of the 4 principal ribonucleotides ATP, GTP, UTP and CTP. These compounds serve important coenzymic functions in the metabolism and are the immediate precursors for ribonucleic acids or RNA, and roughly 90% of the entire nucleic acid content in the cell is RNA, with the remainder being deoxyribonucleic acid or DNA. DNA differs from RNA in being a polymer of deoxyribonucleotides, one of which is deoxythymidyllic acid. We will now turn to the degradation of these compounds. In some organisms, free pyrimidines are salvaged and recycled to form nucleotides via the phosphoribosyl transferase reactions. In humans, however, pyrimidines are recycled from nucleosides but free pyrimidine bases are not salvaged. Catabolism of cytosine and uracil yields beta-alanine, ammonium ions and carbon dioxide, while the catabolism of thymine yields beta-amino isobutyric acid, ammonium ions and carbon dioxide. In order for the rings to be cleaved, they must first be reduced by NADPH. Atoms 2 and 3 of both the rings are released as ammonia and carbon dioxide. The rest of the ring is left as a beta amino acid. Beta amino isobutyrate from thymine is largely excreted. The beta alanine from cytosine or uracil may either be excreted or incorporated into the brain and muscle dipeptides, predominantly carnosine and anserine. The scheme that is now displayed on your screen will summarize the catabolism of pyrimidine nucleotides. Now, let us do a brief session on the disorders associated with the degradation of pyrimidines. We will start with orotic aciduria. This is a rare metabolic disorder characterized by the excretion of orotic acid in urine. This causes severe anemia and retarded growth. It is due to the deficiency of the enzymes orotate phosphoribosyl transferase and OMP decarboxylase of pyrimidine metabolism. Both these enzyme activities are present on a single protein as domains. In other words, it is a bifunctional enzyme. The hereditary form of orotic aciduria is an autosomal recessive disorder. It can be caused by a deficiency in the enzyme UMPS, a bifunctional protein that includes the enzyme activities of orotate phosphoribosyl transferase and orotidine 5' phosphate decarboxylase. It can also arise secondary to the blockage of urea cycle, particularly in ornithine transcarbamylase deficiency or OTC deficiency as it is referred. Increase in orotic acid secondary to the OTC deficiency forms hereditary orotic aciduria, and it can be distinguished by looking at blood ammonia levels and the blood urea nitrogen. In OTC deficiency, because the urea cycle backs up, you will see hyperammonia and a decreased blood urea nitrogen. Intake of a diet that is rich in uridine or cytidine is an effective treatment for orotic aciduria. These compounds provide pyrimidine nucleotides required for DNA and RNA synthesis through phosphorylation. Besides this, UTP inhibits carbamoyl phosphate synthetase 2 and blocks the synthesis of orotic acid. Uridine provides UMP through phosphorylation, which not only replaces the UMP normally synthesized but also inhibits CPS2 so as to decrease the rate of orotic acid synthesis. Replacement therapy has also been successfully used in this disorder, whereby uridine is phosphorylated in vivo to uridine 5' phosphate or UMP by the enzyme uridine kinase. This way, the enzymatic block is bypassed. The presence of this nucleotide simultaneously reduces orotate production through end product inhibition on the pyrimidine pathway. The next condition that we will consider is Reye's syndrome. This is considered as a secondary orotic aciduria. It is believed that a defect in ornithine transcarbamylase, the enzyme of urea cycle, causes the accumulation of carbamoyl phosphate. This is then diverted for the increased synthesis and excretion of orotic acid. Reye's syndrome is a rare but often severe and even fatal illness that primarily occurs in children and adolescents. Children diagnosed with Reye's syndrome generally suffer with excessive vomiting and mental status changes. These symptoms are generally the results of increased intracranial pressure and brain swelling. If untreated and progressive, the disease can be fatal. Even if identified and treated early, some patients will still have progressive disease resulting in death or permanent brain damage. Thus, the illness can resolve spontaneously or progress to coma and death. Unfortunately, there is no absolutely effective treatment for Reye's syndrome. Primarily, the treatment is aimed at decreasing the effect of the metabolic dysfunction. Patients with Reye's syndrome are admitted to an intensive care unit and monitored for a worsening neurologic and metabolic condition. The primary goal is to manage electrolyte imbalances and brain swelling. It is difficult to predict which patients will have a progressive illness. However, some doctors recommend using medications aimed at lowering the serum ammonia level because ammonia is known to be one cause of increased brain swelling. In addition, in some cases of progressive and resistant Reye's syndrome, hemodialysis has also been used to remove toxins that are believed to be partly responsible for the brain swelling. However, the only aim is to keep the patient as comfortable as possible. Let us now summarize the salient points of this lesson. Nucleotides participate in a wide variety of reactions in the living cells, predominantly in the synthesis of DNA and RNA as constituents of many coenzymes and in the regulation of metabolic reactions. Purine nucleotides are degraded to uric acid, the excretory product in humans. Uric acid serves as a natural antioxidant in the living system too. Gout is a metabolic disease associated with the overproduction of uric acid. This often leads to the accumulation of sodium urate crystals in the joints, causing painful gouty arthritis. Allopurinol, an inhibitor of xanthine oxidase, is the drug used for treatment of gout. Severe combined immunodeficiency or SCID represents a group of rare and fatal congenital disorders characterized by little or no immune response. The most common treatment for SCID is bone marrow transplantation. Pyrimidines are degraded to amino acids, namely beta-alanine and beta-aminoisobutyrate, which are then metabolized further. Orotic aciduria is an autosomal recessive defect in pyrimidine metabolism that is caused by the deficiency of orotate phosphoribosyl transferase and OMP decarboxylase. It is treated by a diet rich in uridine and cytidine or by replacement therapy. Reye's syndrome is considered as a secondary orotic aciduria. It is a defect in ornithine transcarbamylase enzyme, a component of the urea cycle. This causes accumulation of carbamoyl phosphate. Thus, nucleic acid components not only play a vital role in the functioning of the cell, but any defects in their metabolism can lead to dire consequences. With that, we come to the end of this module, and this is Dr. Padma signing off. Thank you.